tonight it gives me absolute great pleasure to invite Dr Caitlin Finney to the stage, who is the Senior Postdoctoral Researcher at the University of Sydney and is the latest beneficiary of funding from our charity, and she's going to tell you all about the research project that we are funding tonight.
Thank you.
Thank you.
Perfect. Hello, everyone. It's lovely to meet you all, and thank you very much for having me tonight. I really appreciate the opportunity. So I thought I would start out with just a little bit about me. I'm sure you can hear by the fact that I just said about that I'm definitely not Australian. I am 100% Canadian through and through. Very happy to be here. I've been here for 10 years now and love driving on your roads relative to the Canadian roads, which are always exciting. So I started out my sort of journey in research doing a Bachelor of Honours in Neuroscience at the University of Toronto. So I'm originally from Toronto. And when I was first year undergrad doing my degree, my uncle had a really bad stroke. And his stroke caused him to have dementia. And so he had post-stroke dementia. And I was able to witness firsthand what it's like to have a family member diagnosed and for the doctors to really have no options for that person. This was about 10 years ago now, and so there are even less options than there are now. And I know what that is exactly like, that feeling. And I remember thinking at the time, I was debating between doing research or going to medical school. I was really interested in neurosurgery. And I remember thinking to myself, surely we can do a little bit better than that about having no options for people. And so that pushed me towards doing a PhD. And I ended up doing a PhD in pharmacology and physiology at UNSW, which is why I'm here in Australia. And I looked at a new drug to treat post-stroke dementia. And so this really pushed me on this trajectory of dementia research. I've moved beyond post-stroke dementia now, and I focus a little bit more on Alzheimer's disease. And I'm really interested in precision medicine, so I thought I would tell you a little bit about what that is. There's a growing realization that diseases are caused by interactions between our genes and the environment. And it's this really unique, sort of delicate interplay between these two things that are driving diseases. And they really need to be treated according to these individual features. So lifestyle and environment are particularly hard to study, so I'm quite interested in studying genotype. My background is biomedical science, and so I'm a little bit more interested in the genetic side of things. And so what do we know about the genetics of late onset Alzheimer's disease? To be honest, not a whole lot. We know a lot and not much at the same time. So we know quite a bit about familial or early onset Alzheimer's disease. That accounts for less than 5% of cases. But we know that there are really specific genetic mutations in 3 different genes that are driving that. When we look at late onset Alzheimer's disease, or the most common form, there are hundreds and thousands of different mutations that have been associated with this particular disease. And so what's really interesting to me is we haven't really pursued any research into those variants. Which ones are functional, how can we treat them, and what can we do about it? And so really that's what my research focuses on. So I am interested in integrating a whole bunch of different fields into one at the same time. And so I sort of took some principles from artificial intelligence and from machine learning, and I asked a computer to tell me how likely is it that a particular genetic mutation or an error in someone's DNA is actually driving late onset Alzheimer's disease? And it turns out that we can do that. So I found 2 particular mutations, which you can see in red, my little dot, and these mutations were highly likely to be associated with late onset Alzheimer's disease, almost to the same degree that our familial mutations, these other 2 in the black dots were. And so that suggests that this is a functional mutation, that there is something going on in some people with late onset Alzheimer's disease. And so I partnered with the US, and we looked at a really large cohort of people with Alzheimer's disease. And we found that these particular mutations, which you can see in red, were quite high. There was a large prevalence. It was actually 3 times higher than what we would expect in the general population. So again, suggesting that we're on the right track. The genes I'm looking at are genes in something called ATP5H. Don't worry about remembering that. It's a gene in the mitochondria. So mitochondria are really small little organelles in your cells, and it's decreased in people with late onset Alzheimer's disease. So we know that mitochondria, they provide the energy for the cells, and they're really the powerhouse of your cell. Brain cells in particular need a huge amount of energy to make sure that they're functioning properly. And so maybe it's the case that some people with late onset Alzheimer's disease might actually have some dysfunctional mitochondria, and that could be driven by their individual genetics. So you can see here as well, there's a picture of a mitochondria, and that's what it looks like in real life. So this is really the powerhouse of your cell, making sure that your cell can chug along and do everything that it needs to do in the daytime. I then look at 3D brain models of late onset Alzheimer's disease. So the way that I do that is I take skin samples from people with late onset Alzheimer's disease who do and don't have my particular mutation of interest. So what's really nice about research now and about science is that we can actually take those skin cells and take them back in time and say, skin cell, I don't want you to be a skin cell anymore. I'd actually like you to turn back into being a stem cell. So how you started out in this world as a stem cell. Once I have these stem cells, I can then tell them, stem cell, I'd like you to become a brain cell. And so I can start to grow these mini brains in a dish in the lab, and we call these brain organoids. And this is all derived directly from the skin of people with late onset Alzheimer's disease. This is what they look like here. So they're quite small. They're about 2 millimeters in diameter. And we can really look at how disease is developing in these mini brains and recapitulate what's going on in real life and in real people. We can also do lots of high-level imaging to start to look at the individual cells in there, to really get at a very high-level dissection of what's going on at that cellular level. And so what I do is I also use CRISPR-Cas9 genome editing to correct or insert these mutations to better understand how changing them or modulating them affects the disease development. And so CRISPR-Cas9 is just a really fancy word for some DNA scissors. So you can imagine a pair of scissors come in, they cut the DNA at a particular part, and then I insert either a different mutation or the mutation I'm interested in studying. So I can fix it, I can cause it, and we can do that all with these really fancy scissors. So that's all it is. It's fairly straightforward. And then I examine, exactly, yeah, just do a PhD, you'll be fine. And then we really examine how these mutations affect the development. Like I said, the really nice thing about this is we can recapitulate it in the lab. So I don't have to rely on recruiting people. I don't have to rely on any of this. I can grow it in the lab myself. And then what I'm really interested in is identifying why. And we examine thousands and thousands of molecules all at the same time to really understand what the mutations are doing at that biological level, at that cellular level. And why is this so important? What I want to do is to leverage that data to then determine molecules that drugs can target to really treat late onset Alzheimer's disease in people with these mutations. So I'll go back to my earlier image, and what that really means is that if I acknowledge or identify that someone has a particular mutation, I know what drug to treat that person. And so we're moving beyond the sort of one-size-fits-all approach to really saying we want to look at Alzheimer's disease as unique in a particular person, not equivalent to someone else's Alzheimer's disease, and to really treat that based on these unique features of individual people. And so if we see that they have a different kind of mutation, we can do drug B, or if they have a third one, we can do drug C. And to really sort of bring that into dementia research and to try to shift the treatment of people with the dementias into this personalized approach. And so I obviously want to thank everyone in this room. I'm incredibly grateful and honored to be here and thankful for your support into my research. I also want to thank a lot of people who are helping to make this research possible. So we rely on collaborators, and there's a lot of people who are driving this and trying to really make a difference for the dementia community. In particular, I want to thank Anna Cho, and her picture's up there. She'll kill me, but her picture's up there. She's from University of Sydney, and she's a bioengineer, so she's been helping me build the organoids. And I'd also like to thank Professor Swerdlow and Associate Professor Wilkins. They're both at Kansas University, and they're part of the largest NIH-funded Alzheimer's disease center in the US. And they've been helping me with a really large cohort of people with dementia that we just haven't quite built here in Australia, but one of the things I'm also hoping to tackle at some point. And thank you to the other funders who have also kindly donated to my research. But thank you again for having me. I'm very happy to chat about research, about all sorts of things, about being Canadian, where to get best maple syrup in Australia. I know answers to all of these things. So thank you again for having me tonight, and I really appreciate it. Thank you.